without further ado, John, I think you're going to present ciliarin node first. So let me see, I saw you before, but now I've lost it. There you are, great. Super, over to you, John. Your clock starts now.
Great, so good afternoon, everyone. For those that you don't know me, I'm John Sayer from Newcastle University, leading the ciliarin network. So next slide, please. So in our sort of investigator team, there's myself, there's Pleasantine Mill from Edinburgh and Albert Ong from Sheffield. And then we've got Audrey Hughes, who's our patient representative. Audrey Hughes is a big sort of fish, actually, with the PKD charity. She's like a member of several boards for the PKD charity and has actually worked really hard with us in ciliarin and developed her own patient board just for this project. So it's really been great to have her on board. I also have Phil Hall, our project manager, who's helped me progress matters and help with these slides for today. Next slide, please. So the vision that we set out to the MRC was that we were interested in people with renal ciliopathies. Well, what are they? They are a set of diseases that cause cystic kidney disease, essentially. They're inherited and they cause childhood and adult onset kidney disease. There's lots of patients with renal ciliopathies, but they still form under this umbrella of a rare disease diagnosis. And we really wanted to enhance the diagnosis of patients and provide patient cohorts so that when drugs become available for certain genotypes or certain patients, that we had patient cohorts that were trial ready. And I'll talk a bit more about this later. And of course, we wanted to build the community, build our relationships with academics, with clinicians, with industry, to try and help translation of new drugs for these renal ciliopathies, many of which don't have any disease sort of targeted treatment at all. Next slide. So when we had this proposal, as you know, we had to submit the sort of vision, which was harmonizing the diagnosis, genetics, and cohorts that I've mentioned, but also the three sort of embedded projects with Sheffield looking at the sort of genomic landscape of ciliopathies, Edinburgh looking at cell-based assays and biomarkers of disease, and me in Newcastle looking at sort of translational therapies for this disease. And along the way, we are interacting again with PKD charity, Bartter-Biedl Syndrome UK, and other charities that are relevant to this disease group. And we've interacted again with industry, and actually in this space, industry are coming on board in very interesting ways, actually. So for the first time ever, there's lots of new drugs coming to the market for various forms of cystic kidney disease. Next slide. So we haven't had to set up a registry. We haven't had to sort of pull our hair out sort of trying to do this, because fantastically, there is already a resource called RDR, which stands for Rare Disease Registry for kidney diseases. And actually, if we look at registry data for ciliopathies, which include this condition ADPKD, autosomal dominant polycystic kidney disease, there's nearly 8,000 patients already in the registry. For the pediatric versions of cystic kidney disease, so-called autosomal recessive polycystic kidney disease and nephronophthisis, there's smaller numbers of patients because it's rarer, but actually because it's probably under-recognized and under-recruited into RDR. So we hope to build on this already fantastic national database to enhance and enrich this database and, as I say, develop trial-ready cohorts. Next slide. So one of the fundamental things that we're trying to do in the lab is study these diseases to find disease mechanisms. And our kind of raw materials, if you like, are these cells that get washed out from the kidney into the urine. And so these have become our starting point for multiple sort of research studies. And so one of the things that ciliarin set up at the very beginning was ethics approval to collect urine from these patients to then downstream do omics and drug assays and all the downstream things you've got once you've got a cell culture growing from a patient. they also provide very personalized sort of disease models, which are really, really powerful. And so we've published several papers now using these renal epithelial cells as models for cystic kidney diseases and ciliopathies. Next slide. Yeah, so next slide again. So I'm building into our sort of research structure the clinical sort of side of things. So I see whole families together with my colleagues in pediatrics in clinics at the Freeman Hospital in Newcastle, where we're hoping to see the weird and wonderful rare ciliopathies, recruit them to studies, provide molecular genetic diagnoses for them, and give them answers and give them hope that their rare disease is being worked on by experts both here and around the country. So this model has been really, really successful. Next slide. The involvement with charities, I've already mentioned PKD Charity, that Audrey helping us out. The Ciliopathy Alliance is kind of an umbrella charity that includes lots of rare conditions that are all related to the primary cilium. So again, they're a powerful sort of voice, and actually, are a voice across both the UK and Europe actually for patient advocacy for ciliopathies. So really important that we maintain our links with those. Next slide. So progress, I think we're making great progress. As you say, we're kind of halfway point, but we've got a strategy board, we've got a patient board, thanks to Audrey. We have regular meetings. The research is underway. So I think we're ticking many of the boxes that we set out to achieve. Next slide. The RDR data enrichment. So I mentioned RDR is a great resource, but actually if all the boxes aren't filled in, then it's less good. So we're undergoing a 6-month sort of enhanced enrichment process, employing research nurses to go through the patient data to make sure every box in terms of kidney imaging, kidney omics, and clinical features of the disease are all entered. And again, this points to the fact that we're trying to develop trial-ready cohorts. We've got ethical approval, as I mentioned, with the urine studies. We've got the local NHS trust completely on board. And so, yeah, all that is up and running without any further delays. Next slide. This is the graph of the recruitment to that rare disease group, the recessive PKD and nephrolithiasis. And you can see, if we go to the next slide, that our data enrichment actually is included a big uptick. This was just data from a couple of months ago, but that tick continues to go up as we recruit new patients across the whole UK representing this rare disease. So I'm really, really pleased that this is going so well. Next slide. And then network building. So I'm a part of an EU Horizon consortium that works on ciliopathies as well. So that has been sort of a good sort of synergistic project to work on. We're working with industry. Hybridize, we're an oligonucleotide company. They're less involved now, but we're working with a company called Elox Therapeutics that are designing nonsense read-through compounds. So that's going really well. In terms of engagement, we had our launch events and we've had several big public meetings. UK Kidney Week is the sort of premier UK kidney conference, and we presented CiliaRN at the Edinburgh and Bournemouth meetings. The next meeting is in Harrogate in March next year, where we're also presenting. And then there's been international meetings. Cilia 2024 was in Dublin, the next one's in Rome in 2027. And then we've had regular node and hub meetings hosted mainly here in Newcastle and online. Next slide. So here's some sort of highlights. So Edinburgh hosted a UK Cilia network meeting. UK Kidney Week, Pleasantine Mill, one of our co-leaders, presented at UK Kidney Week. And then we've got myself and Audrey presenting on behalf of the PKD charity at the Kidney Week meeting. Next slide. In terms of scientific outputs, we've published both clinical sort of papers discussing management and guideline, as well as basic science papers related again to mechanistic data from patient-derived cell lines. So I think our outputs in terms of research fish and the MRC are exactly where we need to be. we've widened the recruitment to the RDR group to include all the rarer syndromic forms, because the wording was initially quite narrow to just, a narrow group of conditions. So we've widened this to allow anyone with a ciliopathy that has kidney involvement to be captured as part of this cohort, because we think treatments for these conditions will be relevant to lots of different syndromic forms of ciliopathies. And then one of the most exciting things is the Genomics England pipeline. So I've been involved with Genomics England since the start, so almost 20 years ago. But now we're hoping to get direct data feeds of positive results from Genomics England into RDR, so that we miss the step of manually having to enter in genomic variants. And so we're building this genomic profile of patients within the RDR cohort, because certain therapies like chaperone molecules rely on certain missense changes in genes, and certain therapies like nonsense read-through drugs rely on stop codons. So knowing our cohort and the genomics of our cohort becomes essential. And we've had great success actually in getting the MRC mouse node to make mouse models relevant to our patient diseases. So we've had a mouse model of TOLP3, WDR19, and a different CEP290 mouse made in the last 12 months. And these mice now sat in Newcastle, breeding well and being phenotyped and proceeding to therapeutic studies. So I'm almost at the end. Next slide. There are great opportunities for this rare disease node. So you've mentioned LifeArc already, but the kidney ecosystem is a LifeArc funded project focusing on pediatric rare kidney disorders, and I think we synergize with them. But also the Accelerating Rare Disease trials, LifeArc, is also a great opportunity, and I've started discussions about what a synthetic sort of study would look like with these patient populations, with a view to being very attractive towards industry investment and doing studies here in the UK in patients with renal ciliopathies. There are human clinical trials in situ. So we have MRC funded IMPEDE study. This is using a drug for autosomal dominant polycystic kidney disease. We're about to recruit the first patient internationally for a study sponsored by Vertex, again for autosomal dominant polycystic kidney disease. And there's a new therapy from GSK also for ADPKD that we've recruited phase 2 patients to. So that clinical trial space is really, really opening up, and I'd like to see it open up for the recessive and forms of cystic kidney disease and nephrolithiasis in the future. I think there are good future funding opportunities. So the Kidney Research UK has established a consortium with the PKD charity to fund research in this research space. And pharmacy industry opportunities, we're interacting with the mTOR node, with NATA, with the other nodes, looking for opportunities there. And I've mentioned ELOC's pharmaceuticals with their nonsense read-through compounds. And I think that's a really exciting opportunity. Okay, I think we're almost done. Yeah, so this is my last slide. So this is what we said, that we wanted to create a community. I think we're doing that. A step change in thinking, and that this should be open access to whole genome sequencing for anyone with cystic kidney disease. We've got that. We've got the results coming into a registry automated. We're taking care of rare and ultra rare. We're bringing therapies to the patient for the first time ever, and we're involving patient communities in the design of all this as well. So I think I'll stop there.
Thank you very much, John. That was perfect timing. Thank you very much. So we will go into our second presentation. So this is going to be UpNat. So it's a very busy time for all of us, but I know UpNat, it's been a crazy time for you. Some of us are seeing each other not just on the screen every week, but in person every week, more and more at the moment. So I know you guys had your symposium, but Mina, I'll hand over to you for your 15 minutes starting now.
Thanks very much. So thanks very much for giving me the time to speak. So As before, I think it's worth just going over who's in UpNat and what the management team is. So our director is Haiyan Zhu, with co-direction from Francesco Montoni. And then actually you have a team which includes myself, Alice Davison, Nick Lynch, Carlo Rinaldi, and Catherine Ryan, our project manager. And we're lucky enough to have Ella Whittle, a really excellent postdoc, also involved. As you can see from the list of NIHRs involved and universities involved, this is already an alliance, and that comes from the fact that it's a platform node, really, and it requires skill sets from across the board. Next slide, please. So I'm just going to very briefly go through and remind everyone of each of the major work packages, but I'm going to be focusing on work package 1, which is the one I co-lead with Alice. So the purpose of work package 1 was to generate guidelines for selection of patients and targets for nucleic acid therapies and to facilitate implementation. I'm going to go through the details of the guidelines we're developing, and we're very close to publishing, I'd say probably early January next year. But if we could go on to work package 2 and 3, I'll just take you through progress there as well. So on work package 2, that's focused on preclinical development, and that's led by Haiyan and Nick Lynch from NATA. And UpNat has already started working on specific cases for the development of targeted nucleic acid therapies, and will start progressing the most promising cases that are recommended from work package 1. But in terms of what work package 2 is already doing in terms of development, there's work on a WDR45 target that would be for a form of childhood epilepsy, where NATA is using its capability in terms of preclinical development and tox studies for this case. We're also working with NLORM on the compassionate use of an ASO to treat patients who have inherited mutations in KIF1A. And those patients are at GOSH, but also at other locations. And I think this would allows us to see how we might start adopting antisense oligonucleotides that have been developed external to the UK, how we can identify the patients in the UK who would benefit most. And I think this is a great use case for that, actually. It also requires quite a lot of technical expertise because of the fact that actually patients have to have have to be identified in terms of their allele as well as a specific mutation. Then actually, we're also in work package 2 working with Everyone Medicines to look at a specific exon skipping ASO therapy for NPC1, but that's providing a framework to work more collaboratively with Everyone Medicine in the longer term. And I think that, again, that is really helpful to have kicked off a specific case, but to understand how that works in terms of the whole ecosystem of where nucleic acid therapies are likely to come from. And if we go to work package 3, so that's the next slide. So again, I think there's been a lot of progress made in terms of using AI and deep learning to try and understand where we are actually with nucleic acid therapy drug development. And this is an initiative led by Carlo, who is part of the leadership for work package 3, along with Francesco Montoni. And what they have done is a really amazing piece of work on actually pulling together the efficacy, reported efficacy from ASOs that have been submitted for patent. And that has allowed us to see really the incredible growth of the number of genes that are being targeted and the type of ASO targeting that seems to work best and the technologies that people are using. So I think that's been a really helpful overview of where the field is, but also a direction scientifically as well. So if we can go to work package 1, next slide. So just to remind people of what the vision is for UpNat, it's to provide a roadmap for rapid bench-to-bedside translation of nucleic acid therapies for rare disease. And the key objective that actually I want to think about with work package 1 is that we want to create this cross-disciplinary network. We want to engage researchers in this area, engage sector-based knowledge, improve the access to nucleic acid therapy expertise in the UK, and really develop an interdisciplinary committee of experts to curate guidelines to identify the patients who would be amenable to NATs and who would most benefit from NATs. So next slide, please. So one thing that we wanted to just do was chart what's happening. And even since UpNat was established, I think there's been a real growth in the number of ASOs going into translation and becoming market approved. Most of those, of course, are not N=1 therapeutics, but oh, sorry, can we go back one? But more, but actually are more frequent. And actually, we can see that also in terms of the N=1 space, you know, with Enlorum's activity, there are actually 40 patients worldwide who are now actually receiving N=1 therapeutics with nucleic acid therapies. If we go on to the next slide. In terms of the guideline development, we can see that there's really been quite an increase in activity in this. And I think that's because everybody is seeing the potential of this area. And consequently, there's a real interest in how to get at the right cases as quickly as possible. And whereas most guidelines focused on the variants that were applicable, we can see increasingly that there are guidelines which are actually focused on what other criteria, so the disease that you're trying to treat or the patient characteristics. Next slide. I think it's important to recognize that even if international guidelines are being developed, we work in a UK specific area and there's UK specific infrastructure that really matters. So the UK actually has its own population of more than 3 million people who are currently living with a rare disease. But beyond that, we're also world leading in genetic healthcare, where there's a pioneering WGS services. And I think it's actually really important that, you know, those components are used as much as possible. So there's also things like the generation study and fetal exomes, which mean that we are diagnosing probably earlier in the world than anyone else for some of the disorders that we're seeing. And there are set pathways that are already being established. And I think we heard from Ciliarin that actually that's really important to them too. That infrastructure really matters and the UK is in a leading position for it. If you move on to the next slide, please. So the UK target selection guidelines, what we want to do is make sure that these guidelines are fair, that they are appropriate for the UK, that they are appropriate for rare diseases. And we wanted to make sure that whatever we produced would be accurate, would be fair and transparent so that patients could understand and clinicians could understand why they were or weren't being shortlisted. Sorry, I don't know if you're jumping ahead, but I don't know whether that's me going slowly and you're trying to encourage me to go faster or what's going on.
No, I think there's some gremlins, but I'll keep trying to fish back. Sorry, Mina.
Okay, no, that's fine. So then we also wanted to ensure that they're easy to implement, and that is also, and they're easy to cost in. So if you're going to assess patients, you need to be able to do that in an easy way, which does not require high levels of expertise or can be automated. We decided to do that by actually splitting the guidelines into 4 subsections, focusing on diseases, functional models for an area, the variant itself that might be targeted, and the properties of the patient that might make them appropriate or inappropriate for treatment. Next slide. You can go on, yeah. So in terms of the development team, it actually has been each one of these separate segments, disease, functional model, variant and patient, has been co-led by people across the UK. So Emma Clement, Hannah Tithridge, who clinical geneticist with expertise in working in Birmingham and Gosh, Stefan Sanders and Jackie from Oxford and UCL, and on variants, a team which involves Genomics England through Annalisa Taylor-Tavares and Noor Al-Khatib, as well as Jenny Taylor in Oxford, and at the patient-focused look with Manju Kurian at UCL, but also Carlo in Oxford. So it's really been very much a team effort. If we go on to the next part. The guidelines are structured, as I say, around these 4 core themes. And what we've tried to do is, and those guidelines are now complete and agreed upon across all of us, is to have mechanisms to quickly exclude patients if actually exclusion is the right thing without having to go through a full assessment and full criteria. And so you can see that that's structured here for the exclusion guidelines, and that's the first step, and then scoring in each of these areas. So when we summarize actually how a patient and a disease and an area might score, we're actually doing that on the basis of the disease the patient has, the patient properties themselves, the functional models that are available to us, and the specific variant that the patient carries. So if you want to go on through. We started by assessing market approved ASOs. Now, you can see that the V for variant is always scoring 0. And that is because all the market approved ASOs currently in the market are not specific to a variant and are actually largely speaking targeting in a manner which, if you think with SMA, which is not the genetic cause of the condition, but a modifier. And so actually, and therefore they in fact score 0 on variant and would not be amenable to an ASO according to those criteria. Patients we cannot score because actually we don't have that information on these market approved ASOs, but we can see on functional models, all of them score very highly. And that is actually, you might argue, because the functional models have already been generated since these are market approved. What is maybe most important in this is actually the diseases score highly. So the appropriateness of a disease where there is something reversible and helpful to treat for the patient and could be measurable is actually scoring very highly across the board. The only places where the disease is not scoring highly is in SMA type 0, where there's been an expansion into the use of compassionate use of nusinersen into situations which are normally congenital forms of the disease. And we've also seen that in the case of hereditary ATTR and hereditary angioedema, the reason why disease is scoring low is because alternative treatments exist. And therefore, actually, it's not clear that any further therapeutic would be helpful. If you want to move on to the next slide. So just thinking about how these kinds of scoring systems operate, I'm just going to give you a couple of case examples. So this is a case of a 4-year-old child with suspected Bandos, which was ultimately verified. This child was born following an uneventful pregnancy and a normal development milestones in the early on age. But disease onset started at around 9 months. The main clinical features, which included chest wall asymmetry, intracranial calcifications, developmental delay, and evidence of neurodegeneration, all pointed towards Bandos, and that was suspected. But in actual fact, only one mutation was found in the relevant gene, the C840del. And then, so 840 deletion, sorry. And actually, my team, in fact, on a separately funded project, did targeted long-read RNA sequencing of the locus of CSF1R and identified a second variant that was a splicing variant that resulted in a drop in the most commonly used main select transcript of this gene and the creation of this novel transcript to a high proportion. So over 50% of the transcripts that are being detected from the patient sample have a novel structure that would be expected to be non-functional. And there's a significant drop to 25% of a structure which would actually be useful to be expressed for CSF1R. If you move on to the next slide, please. So how did this particular case score? Well, from a point of view of the disease, it's the disease could be targetable and it's a CNS disorder, which we want to treat. It's severe and life-threatening, and therefore the risk of using a novel therapeutic is there. No treatment options are currently available, so that's all a positive in terms of looking towards an UPNAT score. But Bandos lacks a defined natural history. We don't have clear biomarkers or clinical endpoints in this case. It's also crucially a cause of an autosomal dominant disease, and therefore this child, even if we treated the splicing variant, would still be at risk of a form of hereditary leukoencephalopathy. We also know that functional models, there are some functional models in existence, but we do not have a mouse model, or the mouse model doesn't replicate the phenotype correctly. The variant itself is difficult to target because it's at a canonical splice size region, and an upregulation strategy wasn't felt to be feasible in this case for this time frame. And then actually the fact that upregulation may not actually be sufficient. Then in terms of the patient, unfortunately, this patient had already progressed to quite severe neurological decline, and it was unclear that the patient would be able to survive long enough to be able to tolerate the time it would take to develop an ASO. So actually, we would have to exclude this patient, and the exclusion criteria would be met very early on, in fact, for patient reasons and variant reasons. If you want to move on to the next slide. We then went on to consider another example, Doyne honeycomb retinal dystrophy. This was a case of a man in his late 40s who actually had the main clinical features of mild distortion in his vision, but also central RP and complex atrophy and confluent drusen. He had actually, there had been an attempt to receive treatment through these, sorry, can you go back to the slide before? To anti-angiogenic injections, which were not sufficiently useful for him. And an allele-specific ASO knockdown was developed, and actually this is in publication at present. If we go on to the next slide. So how did this score? In actual fact, this particular case scored incredibly well and was successful. So the retina is targetable, the mechanism is understood, the natural history of the disorder is understood, there are clinical measures that can be used simply, there are functional models in existence, and actually functional assays exist as well. The actual variant itself is eligible for a knockdown. And actually, this scores high because actually an ASO has already been developed against this specific gene. So in actual fact, we could have guessed that this would be a very successful one, and from all the information available. If you want to go to the last slide, or one, I think it's the last slide. So just in terms of future directions, what we're thinking through very carefully is for each form of criteria that we're using to identify patients who would benefit from ASOs, we might think of who should be doing it. So for the patient selection, that has to be the primary clinician looking after the patient, and that's a case-by-case situation. For the disease, we might argue that actually a disease-specific expert is best placed, and you could do that once for a disease. You don't have to repetitively do it for a disease over and over again. We might imagine that functional models is a case of searching the current literature for what's been published on and what's established, and actually that's probably partially automatable. Similarly, for variant selection and the variant criteria, that is very automatable. And I think shifting technologies will be the factor that will make this a little bit harder to implement, and that more and more variants are coming into scope. So I think we are kind of well on our way to establishing the guidelines, and we're hoping to publish, as I say, probably in January with a bioarchive in the first instance. If you go to the last slide. And I just want to, oh, sorry, I'm probably going to overrun. I'll try and go faster. So we had our UpNat symposium, and I just want to highlight that actually that involved a lot of international engagement with Dr. Alban Ziegler coming, Marlon Laufer from the Dutch Centre. There was lots of national policy engagement from Matt Brown and from Kath Bainbridge. And we also had a lot of industry interest from Lajola Labs, Everyone Medicines, and Praxil and NATA. So if you carry on to the next slide. And we have also been focusing on how to communicate to patients why and how nucleic acid therapies might be appropriate and why they would be, and how the process works, because we don't want to raise false hope. And actually, we're in the process of developing patient guidelines with the Genetic Alliance and with Unique to try and develop materials which would prepare patients for thinking about this and understanding the process a little bit more clearly. So we had a great talk from Claire at UpNat, and that was at the UpNat symposium. That was brilliant. If you want to go to the last slide, I think it's the last slide. So this is our wide set of collaborations. I'll move on quickly in the interest of time. And then just to thank the very large number of people who've really been involved in this. So, there's obviously the UpNat core leadership team itself, but we've actually got a massive guideline team. We've got key collaborators from multiple labs, patient assessments that have been done by experts on the specific diseases, involvement of Genomics England as well, and the GOSH Drive initiative, and lots of public engagement and people from the from Hyan's lab too. So I think it's really been a team effort. Thank you very much for the time.
Okay, well, we will go straight into our next presentation. So we have Bill, I think he's going to be the main presenter from the Reolute node. So Bill, your 15 minutes starts now.
Hello, thanks everybody. So I'm Bill Newman. I'm a clinical geneticist based in Manchester, and I lead the rare early onset lower urinary tract disorder Reolute node alongside my colleagues David Long from UCL, Adrian Wolf from Manchester, Neil Roberts from Manchester, Melanie Chan from Imperial, and Mena Clackworthy from Cambridge. And we're working very closely with ERIC, who are a charity specifically focused on children with bowel and bladder disorders. So next slide, please. And next slide. So when we set out on this node, we had a number of things that we were very keen to do. So we were very keen to encourage some of our junior researchers to take a leading role, and hope I can demonstrate that that's very much been the case. We were very conscious that a lot of the work around these rare bladder disorders, lower urinary tract disorders, were focused around Manchester and London, and we were very keen to involve colleagues across the country seeing patients and children in their clinics, and we were looking to see how we could be sustainable beyond the 5-year funding, and thinking about some of the approaches to that. And I'll tell you a little bit about Melanie's work with the RDR registry, which you've already heard about from John. Next slide, please. So we set out 3 work packages. The first, very much a discovery work package. This is built on our work over a number of years, where we've identified the genetic basis of a number of rare bladder voiding conditions, like urofacial syndrome. And we're taking 2 major approaches, one looking at rare, novel, highly penetrant variants in conditions that might be syndromal, like Shinsel-Gideon syndrome, or they could be conditions that purely affect the bladder. And then Mel, building on her work from the 100,000 Genomes Project, is looking at a more common variant and structural variant approach in larger data sets of children with these conditions. And we've also been exploring how we can work with the epigen rare node, to explore if we can look at methylation profiles, particularly on bladder exstrophy. Next slide, please. So one of the things that we set out was to build our recruitment infrastructure across the country for the rare causes of these conditions. And so we're now open in Manchester and London, but also in Newcastle and Glasgow. We're just about to open at a number of other sites, so Liverpool, Sheffield and Birmingham, and we've had conversations with a number of other sites, so quite a good geographical spread across the country. And then Mel has worked with her colleagues Lucy Plum in Bristol and Jo Cloddia at the Evelina in London to set up within the RDR program, which has been established for a number of years, only relatively recently, and within the course of this funding program, been able to set up for both children and adults with structural malformations affecting the kidneys and the lower urinary tract, a particular stream within RDR, which has recruited up to 490 individuals to date. So that's a fantastic resource which we hope to continue will build upon and generate some of the data insights that John has demonstrated within some of the renal conditions. Next slide, please. So as well as discovering genes, we're really keen to work out what they do and what the pathophysiological mechanisms are behind that. This work is led by David in London, with Jess Trundle and with Mena Clapworthy in Cambridge. And so looking at models of bladder fibrosis in a mouse model, generating single-cell transcriptomic profiles and visualizing the lymphatic networks around the bladder are all part and parcel of this program that's been progressing very well, particularly focused on bladder exstrophy, which has been a very understudied condition, and one where the etiology is very poorly understood. We've also excitingly generated evidence that variants in patch1 are relevant to this condition. And of course, you'll know patch1 is a gene important in the hedgehog signaling pathway and is associated with a condition called Gorlin syndrome. These variants are found in a very specific part of the gene, and we've worked with the MRC mouse unit to create a mouse model, which is been developing very nicely. We hope to receive those mice for characterization soon. Next slide, please. And then building on the work that I mentioned right at the beginning, that one of our key discoveries a number of years ago was to find the genes that cause this rare bladder voiding condition, which is unusual in that the children affected also have an unusual appearance, that when they try to smile, it looks as if they're crying. It's called urofacial or Ochoa syndrome. And so Neil Roberts has been driving this work forward for a number of years, working very closely with Adrian, and has shown that this is a autonomic peripheral neuropathy, and has generated a knockout mouse model which recapitulates the human phenotype, and over the last few years has created an adenovirus gene therapy, which works very effectively in mice. And with Philippa Lopez, through the course of this program, has been exploring the effectiveness of that gene therapy in human cells, and looking at, you can see the beautiful figures of transducing those cells, both smooth muscle and neurons, and showing how you can co-culture neurons and smooth muscle to recreate a model in which you can test the efficacy of that therapy. Next slide. And next slide. So a number of papers have emerged over the last little bit. We've got a number of others that have been are in the pipeline, but we have leveraged about £2.5 million as well through MRC, Kidney Research UK, and Urology Foundation grants, which have been very positive. Next slide, please. And we work very closely with a number of groups to have a number of different meetings and conferences where we've shared our thinking. So the picture at the top is a patient, Sebastian from Colombia, who's affected by urofacial syndrome, who is a doctor himself and has set up a network of patients across South America who are affected by this condition. There's a founder variant in Colombia, which was first recognized by Ochoa, who the condition is named after, and he was very inspiring, and we've presented at a number of other meetings, but also to a number of charities, including the Eric charity, and we held an event with John and the team in Siloren up in Newcastle as well. So very keen to see where our programs of work can link with some of the other nodes. Next slide, please. We have an international steering group, a scientific advisory board, chaired by Norman Rosenblum from Toronto, colleagues from Dory Lam from the States, Alina from Germany, Jennifer Rohn at UCL, and Bert Bugu from Turkey, who was the president of the European Urology Society. So a wide international reach, and making sure that our work is very much aligned with international efforts, and that it's relevant to some of the work that's going on from these top researchers in other parts of the world. Next slide, please. So, as I said, right from the beginning, we work very closely with our partners in the charity and third sector and patient groups, and that's been really important to us, and finding out what are the key things that they want us to focus on. And so we've got some co-chaired online patient forums coming up over the next few months, which are going to be led by our local PPIE group and Eric. We've also know that for a number of these conditions, there aren't any specific treatments. And so providing guidance information is really important to make sure that people get access to the right care and guidance. And I think that might be my last slide. If you just flip to the next one. Yep. Thank you very much. Short and sweet, but happy to take any questions.
Thanks very much for that, Bill. So we will move quickly into our next presentation. This is the cardiovascular node. So James, I think we've got you on. I think you're presenting. Yes, perfect. So your 15 minutes starts now.
Thank you. Okay, great. If you want to jump forward to the next slide. Yeah, so I'm going to give a summary of the research projects that we said we do and where we got to, and then talk a little bit about networks. So if you want to hop onto the next slide, I should have put a slide of who's who at the beginning, but there is something at the end, so we'll come to that. Again, I was sort of thinking the audience was mainly people who were already aware of the node, so I hadn't put much background into which diseases we're looking at, but the rare and genetic diseases we're focusing on are cardiomyopathies and hereditary arrhythmia syndromes, and their big impacts because they cause sudden death in young people and are leading causes of heart transplantation. And so, if we want to understand these, we need to have studies in humans to identify genetic determinants of disease. We need natural history studies. We then have experimental systems to understand pathomechanisms and develop interventions. And then finally, we need to test those in clinical trials. And we felt in our area, in cardiovascular space, that what we really needed was the cohort building part of that, and that the UK's competitive advantage lies in the opportunities to gather data from our single provider health system at scale to do 3 of those 4 activities that I just described. And so we proposed 3 projects that would move us in that direction. The first is to establish a register of everyone in the country with a rare and/or genetic cardiovascular disease, and that would be unconsented data. Secondly, we want to build a national genetic variant database. So at the moment, there are 4 English labs carrying out most of the cardiovascular genetic testing. There are labs in the devolved nations, and then there's some testing being done through whole genomes and other pipelines. And there's a lot of telephoning between labs or not telephoning because people don't have time to telephone to try and interpret variants. Have you ever seen this variant? And we wanted that data to be in one place so that everyone could see it, and the quality of genetic testing would go up substantially. And then thirdly, our third project is to build on number 1 by building a consented cohort of patients whose data are more available and enriched, and we defined a particular pilot study for that. So if you want to jump on the first, I'll talk about the first project. So this is led by Tom Lumbers in UCL and Erin as a postdoc in the team. And if you want to go forward one more, this is really made possible by a partnership with the NHS National Disease Registration Service. So here they have a very particular set of directions that gives a legal basis to hold information without consent at scale for patients with congenital anomalies, cancer, and rare diseases. Now, fortunately, I mean, some of the diseases we're interested in are not rare by this definition. If you take, for example, hypertrophic cardiomyopathy, it affects about 1 in 500 of the population. But if you're thinking about genetic therapies, of course, you need to be thinking about individual genes and mechanisms. And as soon as you stratify the cohort in any way, you get down below this rare threshold. But crucially, in fact, this statute allows us to hold data from individuals who have a genetic test indication or an Orphanet code, as well as having just a straightforward less than 1 in 2,000. Also, helpfully, it's not just people with a confirmed diagnosis, but also suspected or at high risk. In other words, family members carrying a variant would also be covered. So if you jump onto the next slide, we've been working with NDRS to build their rare registry and particularly cardiovascular diseases. So their typical model has been to go to centres and say, who have you got with whatever disease it is? Certainly in our space, that's not really deliverable. The sort of entities, the way that NDRS would code these conditions does not match the data that is held in hospital services. There are also too many services around the country, and the way they're structured, I think, is going to make that not scalable. So I think our biggest opportunity that we've made the most progress on is using the genetic testing pipeline. So in England, at least, we can tap into that There are 4 labs delivering the NHS testing. They submit what's called patient level contract monitoring data, PLCM, centrally, and that crucially says the NHS number of everyone who had a test, what the reason for the test was, and at a high level, what the outcome was. So we're using that essentially now to populate the register. There are then, I've put on this slide some of the other routes that one might use. So not everyone gets genetic testing. We can use hospital episode statistics, but not all of our diseases have ICD-10 codes, and those HES codes are only coded for inpatients, and most of our care is delivered in the outpatient setting. So that's not sensitive to pick people up. There is a plan for a national audit that would potentially identify everyone coming through a specialist service at secondary care, and that might be a very robust additional pathway. And we're also working with one of the networks, well, we've got partnerships with several networks of excellence, but one of them is specifically to see if we can mine other sources of records, letters, primary and secondary care records to enrich this. So that is my very brief view of our register. If you want to jump on. The second project is to establish a genetic variant database, and this is led by Kate Thompson in the Oxford Genomic Laboratory Hub, assisted by Giovanni Stefani, a postdoc. And this has been very successfully, we were set out to follow a model that's been very successful in cancer from the CanGene-Canva partnership, led by Claire Turnbull, that I've been peripherally involved in. And if you want to jump onto the next slide, that pathway essentially involves each laboratory that does genetic testing creating a bespoke data set that they upload to a central register, which is laborious. And we thought it's not the most streamlined approach if we're going to scale this out. The laboratories are already very stretched. So if you want to jump on again, sorry, one more. We're trying other routes of getting this data essentially. So as I mentioned before, we can get an index of who's had a test from the PLCM. And then we were trying to enrich that, find different ways to enrich that data with the actual contents of the genetic test result. So the PLCM just says whether there was a diagnostic variant found or no variant found. It doesn't say what the variant was. And so we thought one route would be for the labs to simply deposit their raw data. So VCF is variant call format, is a primary genetic data set. But NGRS aren't really set up to process that. So we've been in discussions with NGRL about doing it or setting up a space in NGRS. We talked about the laboratories submitting the data that they would be submitting to, for example, ClinVar, which has data about reportable variants. If you just want to click one more, the place now where actually this is not a solved problem, is the short answer at this point in the project. So there's a lot of effort now going into mining the genetic variant reports, which seems sort of counterintuitive that you start with structured data, you put it into a PDF, and then you work hard to get it back out again. But it is something that can be deployed centrally without interrupting NHS workflows. So that is still not fully bottomed out, but making very good progress. If you want to jump on one more. So the third project involves all of us really. I'm perhaps leading some aspects of this. To go on again, the challenge really with our rare register is that, as I said, it's the patients are not provided explicit consent, and therefore the data is very securely held in NHS England, but to an extent that makes it difficult to operate with it. So you can apply for anonymized data sets through DARS, but the rare register is not yet onboarded into the DARS ecosystem, though there is a plan to do that. Another way of working with the data is to have an NHS honorary contract. No one in our node yet has an NHS honorary contract after whatever it is, 18 months, nearly 2 years. So they should have. So that is not a quick process. We want to hold genomic data, and the environment is not set up for genomic analyses. Also in these diseases, primary phenotype data is really important, particularly digital imaging and ECGs. And again, the ecosystem isn't really set up to store, let alone process those data types. So our solution to that is essentially to try and, if you want to jump on a slide, to seek patients' consent to move their data into an environment where we can operate on it more freely and provide access to users more freely, essentially a sort of UK Biobank or Our Future Health, but for cardiovascular rare diseases. And we've made a lot of progress here. So we've agreed a partnership now with NHS DigiTrials, who are a sort of NHS solution for contacting patients. And they have, again, the governance in place to reach out to a patient. And we would say, for example, you had a genetic test. We think that might mean that you're eligible for our study. Would you like to visit this website and provide e-consent to join a cohort? And then, so we're not telling them anything they don't know. We're not saying you have cardiomyopathy or you have long QT. We're saying you've had a genetic test, and invite them in that way. And then their data would move into a trusted research environment that is much more accessible, including to industry, for example, for a cost recovery model to make it sustainable. If you just want to jump on one more slide, there are a few key things that aren't in place. So right as of today, we cannot, NHS DigiTrials can't see the rare register because it's not in DARS, as I mentioned. So that is a key piece that needs to join up, but they can see hospital episode statistic data. So we've got some work underway to sort of validate those codes, and we can start this process by writing to people from that source directly rather than from the rare register, while some of the other pieces are joined up. So our ethics has been submitted, but we have the DigiTrials agreement sort of in place in principle and contracting underway. We have the e-consent platform called the Heart Hive built and running. We already have people signed up in the Heart Hive as willing to participate in research. There's about 970 patients enrolled in there at the moment. And another challenge that this may help us with is, or another challenge we face is, we want this to be a UK-wide initiative, but MDRS is an English system. So for the devolved nations, we need to put in place pathways that directly take them to this e-consent route, and then they'll be in a national research study that is not bound to the English governance of NDRS. Okay, thank you. I think that. Oh no, yeah. Next slide. So where we're at now is our pilot funding really has got a sort of 2/3 of the way down. We now need to scale up and get big funding. We've had some very encouraging conversations with the British Heart Foundation, who have indicated they'd be very pleased to receive a substantial bid to support this, but they would ideally like us to have an industry partnership along a similar model that the UK Biobank had for their sequencing. So I think there are a lot of companies who would be interested in either the genetic architecture of cardiomyopathies as an insight into heart failure and things, or natural history of genetic diseases, or interested in finding patients for genotype specific trials. There are currently, I mean, I can think off the top of my head of half a dozen gene replacement therapies for specific genotypes, looking to identify patients, looking to understand the natural history of disease to design trials. So actually there is a commercial need and set of resources available if we can partner. Okay, I'll to jump on again. So we've this has needed a lot of PPIE. Our main partner has been Cardiomyopathy UK, but we run some specific workshops around trying to understand how to do this process and whether it be acceptable to patients. Next slide. I won't go into this in much detail given the sort of 15 minutes. We partnered with a team called Future Better Consulting, who helped us run some workshops, which were really enlightening. They told us revealed lots of enthusiasm for the idea, for unlocking data, for doing what in a way people think the NHS can do anyway, but for really enabling that. There were some concerns, but I think they were addressable, but it was very helpful for us to understand what we needed to be careful about and where our messaging had to be very clear. And they also highlighted some opportunities and bugs on this table for things to think about. If you want to advance one more, we talked a lot about the sort of, well, not a lot, a bit about branding and how, who should be contacting patients. If you're going to get a letter from NHS DigiTrials, should it be from the NHS, a patient charity? Should it be from Rare Disease Research UK? Who might it be? And it was very helpful to understand, how patients perceived universities, hospitals, charities. It was clear that the NHS is, as I said at the beginning, our strategic opportunity. I mean, the NHS is hugely trusted, although people would rather have things come from their local hospital than a sort of anonymous central office. But if we can show partnerships locally, that would be very helpful. Next slide. Yes, and then I just put in here some quotes from people that were very positive and uplifting around, instead of pockets of research done here and there, let's, bring it all together and do it properly. So I was very, we were very encouraged by this. So next slide, please. We've just come into the network and one more. So this is the team at the beginning, and so we already had a pretty big network at the application. Some of those partners on the slide below, you can see a wide range of academic partners and other initiatives. I've mentioned Kate Thompson and Tom as we went through from Oxford and UCL. Stefan Peterson is the head of the BHF, interim head of the BHF Data Science Centre within Health Data Research UK. Claire is our manager and coordinator for the node, and Rachel has been is in my team and has been really instrumental in a lot of the ethics around the cohort study component. And one more, I think this is my last slide, just to focus on some of the main networking things that we've done. I mean, I've mentioned Cardiomyopathy UK. They've been a really key partner all the way through, although they don't cover all rare diseases. Cardiomyopathies are numerically the largest slice of the pie, and they're a very active and supportive charity, and they've been really willing to help to give us their ideas and to help us link up with patients. The BHF Clinical Research Collaborative is largely a sort of dissemination network. They likewise try to link patients, researchers and clinicians up, and they've offered us some support with that. A big partner has been, a new partner is the British Inherited Cardiovascular Conditions Society. This is affiliated to the BCS, British Cardiac Society. And they have historically had an annual conference, which has been quite a clinical affair, one-day clinical affair. We've now partnered with them and we've grown that into a 2-day parallel stream meeting, which is for the research community as well. We've added in a young investigator competition, much more sort of research orientated content, and that's I think going to be a great partnership because it already has the network of clinicians we want to be meeting, but now we're trying to draw that community together with fundamental scientists. And finally, you'll all be familiar with TRCs, the NIHR's Translational Research Collaboratives, which are themes that are supposed to link up all the biomedical research centres and other NIHR infrastructure around the country. In cardiovascular space, that is, the TRC is actually called the NIHR BHF Cardiovascular Partnership, because it also links in BHF accelerators and centres of research excellence. And they have 10 themes, partnership themes that are identified as research priorities, and we have just this month been applied and successfully been nominated to be their sort of newest theme, which is very good for us. It means that all of the BRCs now will be oriented towards being able to say how they are feeding into this theme, as well as some of the others that have been identified as strategic priorities. So we're very pleased to have that additional support and opportunity to grow the network. So I think I'll leave it there. Thank you. And thank you to Tom and Kate and Claire, and very happy to take any questions.
Thanks a lot, James and colleagues. Go into our last but not least of our nodes, which is the lipidomics and metabolomics. So, Bill, your 15 minutes starts now.
Okay, thanks very much. Okay, so I'm Bill Griffiths in Swansea, and I'll give you an update of what's been going on in our node. So next slide, please, and next one again. Okay, so our initial core partners in the node were us in Swansea, then Cardiff University and the University Hospital of Wales, UCL in London, and St Mary's Hospital in Manchester. Next slide, please. So our initial aims, or our aims of the project, really are to speed up the diagnosis of rare diseases using metabolomics and lipidomics methods that are really currently only available in the research labs. And then to translate these methods ultimately into NHS metabolics labs. And besides diagnosis, we're also, besides diagnosis, we're also monitoring response to experimental therapies. Our initial focus really in the node is on lipid-related pathologies, as that happens to be our real expertise. Okay, next slide, please. So I'll just walk you through our technology really. If we go from the left panel to the right, so we handle mostly plasma or serum. Sometimes it's urine, occasionally CSF. So we take our sample and we extract the lipids and the metabolites. And then we go to the next panel where it says LCMS. We then separate out the different metabolites via LC, liquid chromatography, and then we identify them and quantify them using mass spectrometry. We then move to the middle panel, and the output from the mass spectrometer is a number of peaks. And as a first approximation, each peak is a different metabolite, and the height of each peak is proportional to its concentration for example, in the blood. We then get a list of metabolites and their quantities. So then we move on to the right-hand panel. So we then put this data onto pathway maps. So this is a map of how different metabolites one is converted into another in the body. So different metabolites are in the blue squares, and then we have arrows which correspond to enzymes which show the conversion of one metabolite onto another. So we put our concentration data that we got from our metabolomics experiment onto this pathway map, and in the best cases, the disturbance in the pathway map will allow us to help make a diagnosis. So if we move on to the next slide. So when perhaps, go back one, sorry. So when perhaps we have a defect in an enzyme in a rare disease, so as I've got the red line there, if we have a defect in an enzyme, then the substrates of that enzyme will increase in concentration and the products of that enzyme will decrease in concentration. We're measuring all of this in our metabolomics experiment so we can really help in making the diagnosis. So next slide, please. So at the moment, we're receiving samples from quite a number of collaborating sites in the UK. So we get a lot of samples from the University Hospital of Wales in Cardiff. Then from the south of England, we're getting samples from Southampton. We get a lot of samples from the London area. Then from Cambridge, Birmingham, and then in the north, we're getting samples from Manchester and Salford, Sheffield, Bradford, and Leeds. Most of the samples we received, really, they're sent to us for diagnostic purposes, see if we can help the clinician get a diagnosis. Sometimes they are for actually us to try and monitor experimental therapies that the patients are on. And occasionally some of the samples we get, the diseases are really ultra rare. So it's really just to try and learn more about the biochemistry. You see at the moment we're missing out on samples from Northern Ireland and Scotland, so that we're aware of that. So we know we have to reach out more to our colleagues there. Okay, next slide, please. So some of the rare diseases that we're actually looking at. So on the left, I've got a list of some of the ones that we're looking at in terms of diagnostic purposes. So these are, as I said, these are ultra rare. So if we take the second one there, lathosterolysis, the prevalence of that is about one in a million. And the next one down, hereditary spastic paraplegia 5A, that's between 1 in 100,000 and 1 in a million. So these really are rare diseases. For therapy, we're looking at experimental therapies from Wallman's disease and for a couple of patients with lathosterolysis. In terms of biochemistry, as I said, this is where we've got ultra rare diseases. So the first one there, dihydrodolicol diphosphate synthase. There are only 6 families in the UK with where this disorder is found in the children. We're also looking at some of the more common rare diseases. So we're actually doing biomarker studies for Huntington's disease and also motor neuron disease. So next slide, please. So I'll just give you a very short case study of one of the diseases that we can diagnose quite well. So as I said, I mentioned this before, this is hereditary spastic paraplegia type 5A or SPG 5A. So the genetics were uncovered in 2008, and the disease results from pathogenic mutations in the gene CYP7B1. So this codes for an enzyme called CYP7B1. So this is an autosomal recessive disease, as I said, with an estimated prevalence of something between 1 in 100,000 and 1 in a million. So when the clinician suspects a patient has got SPG5A, of course, he will order a genetic test. However, of the 639 known mutations in the CYP7B1 gene, actually 228 of them are variants of uncertain significance. So the genetic test may well bring up a variant of uncertain significance and really equivocal data. So the clinician won't really know whether the patient has got SPG5A or not. So then he has to order a biochemical test, but there aren't any biochemical tests in the NHS. So this is when the samples actually come to us. So next slide, please. So this is one of the easy cases to actually make a diagnosis because we've got a defect in an enzyme. So we've got that red stop sign. So the substrates of the enzyme are elevated while the products are diminished. So this is something we easily measure. Next slide, please. So I'm a chemist, so I have to draw these pathway maps with some chemical structures. So the CYP7B1, the defective enzyme here, appears in the bile acid biosynthesis pathway, which is one of my favorite pathways. That's why I've drawn it. So if a patient's got a mutation or a pathogenic mutation in CYP7B1, then the enzyme's not going to be working. So the substrates, that's 26HC and 3-beta-HCA in red there, should go up. So then, next slide, please. If we look at the plasma from a healthy person and we do our metabolomics experiment, we see a very small peak for 26HC and a small peak for 3-beta-HCA. However, when we get a sample from a patient with bona fide SPG5A, next slide, please, you can see the peaks for these two metabolites go up. So the test tells us that the patient really has got SPG5A. His variant of uncertain significant is pathogenic. So SPG5A seems to be perhaps it's more common than is actually first thought. And we've actually received in the last 6 months 5 SPG5A patient samples, 3 from London. And actually all those 3 patients now may well go on to experimental therapy with statins, and then we can monitor the response to the treatment. If the treatment works, we'd expect these 2 metabolites, 26HC and 3-beta-HC, to go down in subsequent samples. Another patient sample we received quite recently, he came from the Cambridge area, and he was diagnosed with a form of paraplegia at the age of 40. However, now he's 80, and we've now finally got a diagnosis for him. As SPG5A runs in families, of course, this is important for his family. Okay, next slide, please. Okay, PPIE. So in Swansea, we've actually got a very helpful group called the Patient Experience and Evaluation Research Group, and they come to the university. It's a group of non-academics, non-clinicians. They come to the research group. They come to the university once a month, and then we have an opportunity to meet them, discuss our results, and discuss our project. And you know, for those of us who are laboratory scientists, it's really valuable to get a view from those who are really not in our field. We've also been attending a number of patient group meetings. So we've attended the family days for Huntington's disease, motor neuron disease. We've attended the leukodystrophy charity community weekend, the MPS community weekend, and also the NCL BDRSA family conference. So we've been going to these family meetings, presenting our project really, and getting great feedback from the families. Okay, next slide, please. More formal meetings. We presented at the Festival of Genomics in London last year, and then at the International CTX workshop in Kyoto, and then the following meeting, which was the International Conference of Inborn Errors of Metabolism. And we've also presented the Welsh Motor Neuron Disease meeting, and we sponsored the inaugural Welsh Rare Disease Research Network event a couple of months ago. Okay, finally, next slide, please. We've got two publications which I inadvertently omitted, and we've also got three preprints on medRxiv. Next slide, please. Grant application. So we helped Claire Hart get an NIHR fellowship. So Claire is in Sheffield, and she'll spend some time collaborating, coming down to Swansea and working with us. We've got some money from Healthcare Research Wales as part of the Advanced Neurotherapy Centre, and where we'll be trying to monitor response to advanced therapies. And then we've got money for looking at biomarkers for ALS, motor neuron disease, and also for looking at biomarkers of Huntington's disease. And we've got some grant applications in at the moment, and also in the last year or so, we've set up new collaborations, one with actually Dave in Newcastle looking at cholestasis, and also we're hoping to set up a collaboration with Genomics England, where we can tap into some of their rare disease patient samples. Okay, next one. Next slide, please. Finally, the ultimate goal of our work really is to actually translate our methods from the university lab into the NHS metabolic labs. So in the past, we were working with two instrument manufacturers, Thermo and Shimadzu, to try and put our methods onto their instruments, which are actually in the NHS labs. And now we've just started another collaboration with another mass spec manufacturer, Waters, who are in Manchester, again to put our methods onto instruments which are used in the NHS labs. And then next slide, please. My final slide. So this is a list of the various team members in Swansea, Cardiff, UCL, Manchester, and Claire Hart, a great collaborator in Sheffield. So that's my talk today. I guess that maybe I was a little bit quick, but I'm sure no one minds that.
That was great, Bill, and it's always good to hear about all the work you're doing. I mean, it's a perfect example of where you take, individuals with expertise which may not, you may not maybe you have, but you may not your whole life have labeled yourself a rare disease expert, but you come along with, this, the amazing chemistry background, and you apply, new solutions to a rare disease challenge, and this is what we get. Any questions?
It's actually it's amazing when it actually works. When it works and you get a diagnosis of somebody who didn't have it for 40 years. That's the buzz you get from that is amazing.
Yeah, absolutely. Great. So we're getting towards the end of the year, but we're not quite winding down yet. Still lots to do and some exciting plans to start 2026 as well. So thank you all for joining today, and see you all again soon. Bye.
Thanks, everyone.
Bye. Bye.